Today we're going to be talking about protein synthesis. I just want to state right now that I am presenting this to my partner Nicholas and my dog Indiana. She's currently behind the camera right now or to the side of it, but if she happens to walk in front of the camera, I do apologize. She's unpredictable and she's also really excited about protein synthesis. So again, if she walks in front of the camera, we'll just keep going. We're going to start talking about protein synthesis today. First things first, we need to talk about DNA. In order to understand protein synthesis, we need to understand DNA. So we're just going to get a little bit of background information on that. What is DNA? DNA is a self-replicating material, and it is present in pretty much all living organisms. It is the main constituent of chromosomes, and it's also the genetic carrier, the carrier of all genetic information. When taking a look at DNA, we know that it's composed of 4 different nitrogen bases, and they are, to start with, adenine. We represent this using the letter A. We also have guanine, and we use the letter G. We have thymine, and to represent that, we use the letter T. And then we finally have cytosine, and of course, to represent that, we use the letter C. The rule to follow when looking at DNA is that A is going to bind to T using a double bond, and C binds to G using a triple bond, and you can see a diagram here to show a little bit about that. Every sequence uses, using the 4 bases, the 4 base codes can be translated to create any protein in your body. We use these codes in protein synthesis. We also call them codons. So codes or codons, I use those words interchangeably, or you can. And yes, every sequence, when we take a look at the sequencing to create proteins, we use those 4 base codons. In DNA, we have, in DNA, we have 2 sides, side 1 and side 2. Side 1 is the template strand, that's where we get the information from, and side 2 is the complementary strand, and that is where we copy or replicate the information using the other code. So if on the template strand we have an A, then on side 2 or on the complementary strand we're going to have a T. If we have a C, we're going to have a G. If we have a G, we're going to have a C. If we have a T, we're going to have an A. So basically just using those 4 different codes. And then again, another diagram here to show what the original looks like, and then a copied DNA strand. And this is what we're going to use in protein synthesis. So that's our little bit of information about DNA. So now, moving on to protein synthesis, there's 2 main steps in protein synthesis. We've got step 1, which is transcription, and step 2, which is translation. Step 1, transcription, is the process by which DNA is copied or transcribed. And then step 2 changes it from the DNA language to the protein language. And you can kind of use the root of the words to understand that. Transcription, you're copying something, you're transcribing something, you're writing something down. And then in translation, you're translating it to a different language. So you can think about it from translating French to English, or translating from DNA to protein. DNA is the blueprint, so we can use that to figure out what we're going to do, but everything is made up of proteins. And protein is made up of individual amino acids that join together. There are 20 amino acids in your body, or 20 amino acids create your proteins. 12 of them you make, and 8 of them you have to eat through meats, through other, things you consume them. The question here, and what we're going to learn here, is how do we make these 12 amino acids? That is through protein synthesis. So the first step, we have transcription. We're just going to go into a bit of detail here. In transcription, the DNA separates and unwinds with the help of RNA polymerase. And this is, we copy it from the template strand. So using the template strand, we unwind it and we create the mRNA. The mRNA leaves the nucleus and enters the cytoplasm. I apologize, my dog is chewing her bone, so that's what the weird sounds are coming from. mRNA, messenger RNA, leaves the nucleus and enters the cytoplasm to go find a ribosome. In translation, the messenger RNA code is translated into amino acids to create a functional protein. Once it leaves the nucleus, the mRNA finds a ribosome, and the ribosome basically reads the code, and it advances through this mRNA until it reaches the start codon. And I have a slide here explaining the different codons. Start codon is always AUG. Then we've got the tRNA, and this contains the anticodon, which would be the opposite to the codon on the rMNA. And when it finds its match, it drops off an amino acid and leaves. When the ribosome reaches a stop codon, there's 3 different ones. We've got UAG, UGA, and UAA. The ribosome falls apart, and then the mRNA is released and it goes back. The amino acid folds into its protein shape, and then it's transported. So we've talked about mRNA, we've talked about tRNA, but what is RNA in general? It's similar to DNA, but it's single-stranded, and therefore it breaks down over time. And in RNA, instead of thymine, it has uracil, and we use the letter U. Therefore, in RNA, A is going to bind to U, C is still going to bind to G. So when looking at when we were looking at like the mRNA and tRNA, if mRNA has an A, then in the tRNA, the anticodon would be U and not T. U and G and C still stay the same. So again, a little bit more about mRNA and tRNA. mRNA is the type of RNA that is the messenger, M stands for messenger, and it carries the message from the DNA which is located in the nucleus to the ribosome, which is located in the cytoplasm. mRNA is read in 3 bases called codons. So when you have a bunch of different bases, you read them in groups of 3. tRNA is the RNA that is the transporter. T stands for transport, and the anticodons from the tRNA match and drop off the appropriate amino acid when being read by the ribosome. Codons, again, a codon is a group of 3 bases. The start codon would be AUG, and then we have the 3 codons, or the 3 stop codons, which are UAG, UGA, and UAA. So I just have a little diagram here to kind of explain it a little bit better. This would be the DNA. So we've got the template strand here, and then we have the complementary strand. We take this complementary strand and cut a chunk out of it, which then creates our mRNA. And then when we go over here, this black circle is representing a ribosome. I know it might be a bit hard to see on the camera there. Basically, wherever there was a T in the DNA or in the complementary strand, we replace it with a U. And what the mRNA is going to do is it's going to look for the start codon, and here we can see that we've got AUG, that would be the start codon, and from there it starts reading the code in chunks of 3. So it starts with the AUG, the transport tRNA drops off the protein, the matching amino acid. Then it goes on to the next group of 3. Here we have CUC. In this case, I've given it a star. That's the second amino acid. Then we have a third group of GUA that translates to a certain code using the tRNA, drops off a amino acid, and then finally we get to UAA, which we know is the stop codon. There are no amino acids to match up with the stop codon, so that's when the messenger RNA knows that its job is done and it can leave the ribosome. Sorry. There we go. Now we're just going to talk a little bit about mutations and what it looks like when they occur in protein synthesis. So mutations happen for 2 reasons, and we're talking specifically about single base mutations on DNA. There's other reasons that mutations might happen, but we're talking specifically in protein synthesis. So the first reason that a mutation would happen is when there's a mistake in making your DNA. that kind of first step there. And then there's also environmental factors, so carcinogens, or also known as cancer-causing agents, which disrupt the DNA. There's just a chart here to kind of outline the two different types of mutations. So the first one would be point mutation, which is also sometimes called substitution, and then we have the frame shift mutation, which is can be called a deduction or an addition. So looking at point mutation or substitution, it occurs when there is a base gets substituted for a different base. So for example, instead of a C, there's a substitution of an A. And it would only potentially alter a single amino acid in the chain, a single group of 3, because you haven't removed anything. It's just going to affect one amino acid. The other ones are all going to remain the same. Then we have a frame shift mutation, which is much different. Again, it's called a deletion or an addition. And as you can see in the little chart here, that's when an extra code, or an extra base is put in, or a base is taken out. And what that does is it shifts the entire frame of the codons, resulting in a mutation. So in the insertion, your groups of 3 are totally thrown off because there's an extra one added there. And again, in deletion, if you take a base out, all of the codon groups are completely thrown off. There's 2 ways to express mutations. The first way is the missense mutation, mutations that change the protein shape, causing it to work less effectively or not at all. That is what you would see when you have a deletion or an addition. Doing that changes it completely, and most of the time it's going to just make the protein not work at all, or make that piece not work at all. And then we have the silent mutations, and this is what you would see when there is a substitution, because although the amino acid is different, it still works. It's just now completely different amino acid. So these are mutations that occur and have changes in the DNA bases, but the amino acid stays relatively the same, and those would be the silent mutations. And that's it. Thank you.